Dr. Howard Salis at Penn State, well, has got to be one of the foremost leaders in supplying the synbio community with tools and models for de novo programming and predicting gene expression, which has just hugely facilitated the design and tuning of our synthetic genetic systems in bacteria over the last decade. From the seminal RBS calculator in 2009 to the Operon, promoter, and CDS calculators and half a dozen others, the Salis Lab has been doing God's work for the field. And we like to joke that you can look at the DenovoDNA.com job submission leaderboard to see how much synbio is being done at any university. And Dr. Salis also stands out in his user support and accessibility to the community on his Slack, a lesson to tool developers out there. Let's welcome Dr. Salis.
Yeah, thank you very much for that wonderful introduction. I'm very delighted to have the opportunity to speak with you all today. We're a small enough group that at any point in time, if you have a question, just ask. We can make this more of an intimate setting. Today I want to talk about more along the lines of, where do we go from here, right? We've developed, models and design algorithms for making predictions about how to engineer genetic systems. How do we take all of that and develop a engineering discipline for biology? And how does that even work? Okay. And so I just want to start off with an introduction. That's something that's interesting for people who do know what synthetic biology is. And that is, typically when we start off with a project, we specify an objective. And here I'm giving an example where the objective is to treat a metabolic disease in people called hyperammonemia, there we go, which is the toxic accumulation of ammonia in the blood caused by liver disease or a genetic predisposition. And so you can actually engineer a probiotic bacteria to consume ammonia in the gut. And there is a way that that consumption process can actually lower the accumulation of ammonia in the blood. And how do you do that? Well, it's a three-step process. You design a multi-enzyme pathway to convert ammonia into arginine. You design regulatory genetic parts to control the expression levels and concentrations of those enzymes in order to consume the most amount of ammonia. And then you design a biosensor that turns on that pathway only when that probiotic bacteria is inside the human gut. And you can do that by sensing the absence of oxygen. Okay, the human gut is an anaerobic environment. And so this is not just some idle application. This has actually been built and tested and even clinically validated by a company called Synlogic. And it's an example of how the synthetic biology field is taking what we know in terms of engineering principles and actually applying them to develop new therapeutics, new applications, for affecting people around the world. And so there's a good part of this introductory example that this genetic system that was engineered by Synlogic, it does work. It consumes ammonia in the gut. It is perfectly non-toxic to healthy people. A healthy person can take this engineered probiotic bacteria. It's E. coli Nissl 1917. They can tolerate it extremely well. They've figured out the optimal dosage for such probiotics. But during those same clinical trials, they did identify that the design of their genetic system was not good enough to consume enough ammonia for people with diseased livers. So if you have a diseased liver, you're not able to metabolize that ammonia in your blood at all. The concentrations really do rise very precipitously and to a toxic level. And the design of their probiotic bacteria did consume ammonia, but just not enough to treat the disease in these diseased patients. Okay, and so that's an example where the concept is there. The concept and the proof of the principle works. And now we get to the details, and we have to optimize the system to make it work really, really well for the intended application. Okay, and so that is perhaps the difference between early synthetic biology, where proof of principle was all we needed, to now in the future, when you want to engineer your system to work to solve a problem, for a specific objective, you actually need it to solve that problem. It actually needs to perform at a very high level. And so this is not just, synthetic biology is not just for engineering probiotic bacteria. I like to say that organisms are the ultimate catalyst for matter, energy, information conversion. And you can do lots of things, from developing diagnostic assays to manipulating the soil environment, the aquatic environment, scaling up the production of biorenewable chemicals, whether they're materials or fuels or pharmaceuticals. You can do a lot by engineering an organism, but only if we are able to harness all that potential. And of course, there is an economic opportunity here with a sizable positive impact on human society. And so there are now hundreds of thousands of companies springing up that are leveraging biotechnology for solving all sorts of valuable problems. Some of which are trying to replace our consumption of petrochemical feedstocks with biorenewable feedstocks. Others just to mitigate climate change by breaking down plastics and developing carbon neutral approaches to making chemicals of interest. Others are focused on the therapeutic area, developing new therapeutics for cancer, for metabolic diseases, infectious pathogens, and so on. And others are in the food industry, developing plant-based or non-meat-based food products. Okay, there's tons of applications. And so the question that I ask my lab members, we're not really invested in the success of any one of these companies. Our objective is to ensure that each and every one of these companies can overcome the technical bottlenecks to their success. Because in order for synthetic biology to be a mature engineering discipline, we need a mature ecosystem of companies that are developing innovative products. And that's true for all the engineering disciplines. So what are the challenges that we commonly face in synthetic biology? How do we overcome those challenges through predictions and design? That's what I'm going to be talking about for the next couple of slides. So first off, how do we engineer a metabolic pathway or a network without an astronomical number of genetic changes and measurements? We don't want to necessarily go the full brute force approach. And in fact, a full brute force approach is not possible. So I'm going to give you examples of why it's not possible. So first, we can start off by writing down all of the enzymes responsible for converting a substrate like glucose into a product like a specialty chemical, whether it's a biodegradable plastic or a fuel or a pharmaceutical. Now, if you actually do write down all the enzymes from substrate to product, as well as cofactor regeneration and production of biomass, you're going to get a long list. And here I've just written down 18, but in fact, there are more than 18 enzymes involved. But already with 18, you have an 18-dimensional space that you are operating in. You have 18 targets where if you change the expression levels of those 18 enzymes, you're going to be affecting how well the organism is converting the substrate to the product and self-replicating. And importantly, the expression levels of those enzymes in bacteria can vary anywhere from 0, where you knock out the enzyme, all the way up to like 25% of total protein content. And that range is on the order of 1,000,000-fold. So you have a tunable knob for each one of the enzymes in your system of interest, and you can vary the expression level of that enzyme across a 1,000,000-fold range. That's crazy. And so how do you figure out the optimal numbers, the optimal amount of enzyme expression levels to maximize the conversion of substrate to product while keeping the organism happy and self-replicating, while not running out of cofactor, and of course, satisfying other design criteria? And so to get more into the nitty-gritty, we think about, engineering genetic systems. That really means engineering gene expression and transcription, translation, messenger RNA decay rates. And so if you start to enumerate all the different DNA sequences and all the different ways that you can design promoters that control transcription rates, design ribosome binding sites that control translation rates, design messenger RNAs that control their decay rates, it's a lot of ways, right? You end up with a astronomical number of possible choices that we need to make when you go to design a particular genetic system, and the outcomes are quite disparate. So depending upon your choice of DNA sequence, you might vary transcription rates by 100,000-fold, translation rates by 100,000-fold, decay rates by 20-fold, and so on. And so depending upon literally the choice of DNA sequence for our genetic system, you can get very different gene expression levels, which will then, of course, influence how the organism behaves, responds, and performs in your application of interest. And so we need to develop an engineering discipline that allows us to go from specific objective to designed genetic system that has been optimized to perform at the desired level. And of course, this idea, this concept, has arisen before in other domains. I'm going to use chemical engineering because I was trained as a chemical engineer. And so in the early chemical engineering discipline, the field was all about oil extraction and figuring out what to do with all that oil because it was so cheap and plentiful, okay? And very early on, they figured out that you can make aspirin out of oil, you can make gasoline and diesel out of oil, and you can make plastics out of oil. Now, of course, 2 out of 3 of those things we perhaps don't need anymore. We'll still need aspirin in the future. But hopefully we get rid of petrochemically sourced plastics and gasoline and diesel in the future. But this, in going from an early, emerging discipline to a mature discipline, we also figured out the chemistry of catalysis and how to convert, precursor chemical products into fine or upgraded chemical products, as well as a whole range of other, physics and chemistry, ideas, including transport phenomena, heat transfer, mass transfer, and so on. So how do we do the same thing with synthetic biology? What are the core topics that we need to, really understand very well in order to go from an objective to an engineered genetic system that performs really well? And so what we really need are not just one, but a whole toolbox, a whole layering of sequence complete biophysical and system level models. We need the ability to make predictions about what our genetic systems are going to do before we construct them and put them into an organism. If we don't have those predictions, then we are flying blind and we're wasting time, okay? And what do I mean by sequence complete? I mean that our models should be able to accept any DNA sequence and then correspondingly predict their transcription initiation rates. They should be able to accept any messenger RNA sequence and correspondingly be able to predict predicted translation initiation rates, and so on for every possible gene expression process inside the cell. So this is not a small endeavor. This is a pretty big endeavor. That's why I like to say it's a discipline. It's not just one paper or one lab. It's all the labs, the entire field has to work together to make this happen. But we have some early examples like the RBS Calculator where it is possible to make these predictions. Okay, so after you have this ability to make predictions about what your system is going to do before you construct it, you then have the ability to invert the model and then to ask the question, okay, what is the optimal DNA sequence that will give me what I want, that will actually solve the problem and solve carry out the specific objective. And this is, sequence optimization. So you would like to maximize criteria that you want. So for example, if you want a promoter with a targeted transcription rate, then of course you can design a promoter sequence to give you that transcription rate. But also we should recognize that there are various things in our genetic system that we don't want and that we want to minimize. For example, we do not want a cryptic promoter inside a coding sequence, because that cryptic promoter will mess up our ability to tunably control transcription rates in the system and could, of course, change the messenger RNA levels in a way that are difficult to predict. And so not only do we want to maximize the things that we want, we also have to minimize the things that we don't want. And we don't have unlimited choices. We have, for example, a set of constraints. For example, if we specify we want to express certain proteins, we of course cannot change the amino acid sequences of those proteins, at least at this level. Okay, third step, and there are only 3 steps. I'm simplifying it greatly, but we really need to go bottom up to a great extent, in that once we have models of gene expression and system regulation, including expression dynamics, sensing, genome scale metabolism, growth, stress, toxicity. Once we are able to predict the behavior of one cell, we then have to go to multicellular systems and how they interact and how they grow together and perhaps perform biofilms and whatnot. And then those multicellular systems, of course, exist in an environment where heat and mass are transferring, and that's where transport comes in. And then finally, if our end goal is to produce various products, we should also be thinking about how to separate and purify those products. And those thoughts should happen at the beginning of the project, not at the end. Okay. And so this, again, I drew it as a pyramid, and this is not one thing that one lab can do. This is a whole field, a whole discipline of effort combined together. And then it comes, the questions then pop up. Well, how do labs work together to build an engineering discipline? Well, we have to be all speaking somewhat the same language and using the same terms to describe things and to measure stuff, okay? And therefore, as engineers have figured out, it is helpful to come up with a common language. And now I'm of the opinion that that common language has to be based in physics and chemistry, that we really need to be discussing the biophysical interactions, the biochemical interactions. We should be well versed in thermodynamics and statistical thermodynamics and kinetics, and we should be thinking about resource allocation, which is a class of math problems. Okay, and so if you were to, in the future, if you were to major in synthetic biology and get a bachelor's degree in synthetic biology, you might have courses specifically on these topics, because they become so important to understanding how cells work. Mandalorian reference. Okay. All right, so what's our progress so far? So far we've developed predictive biophysical models for translation initiation and translation coupling, for ribosome drafting, which is a non-equilibrium phenomenon, for how ribose switches work and regulate translation rate. We have a new promoter calculator that is able to predict transcription initiation rates. We're also developing models of messenger RNA decay. We've also developed models of CRISPR-I regulation, and of course, multi-enzyme metabolic pathway fluxes. Okay, so which is really chemical kinetics. Okay, so of course, the next step is really to start combining models together, because it's not enough just to have a model of this and a model of that. What we really need is a model of everything, one integrated model. And so how do we do that? And so again, we're making increasing efforts towards that overall goal. One of those efforts is in developing the Operon calculator, where we combine multiple of our models together to design genetic systems. And here you can specify multiple design objectives. So you can have your targeted transcription rates, targeted translation rates, and so on. And then you also specify the things that you don't want. like you don't want internal promoters, you don't want internal start codons that are highly translated, you don't want terminators inside of your coding sequences. You want to make sure that you can actually synthesize your genetic system as fragments. You want to remove repetitive sequences to make your system more evolutionarily robust. And of course, for cloning purposes, you often want to exclude restriction sites. And so again, with modern optimization algorithms like genetic algorithms and so on, you can actually develop very fast optimizers, and you can make predictions about what that final design is going to do before you put it into the organism. And so so my lab has two aspects to it. We develop these models, make them universally available, but then we also apply them to engineer systems with specific applications. And we've engineered portable power supplies for cross-species engineering, extra long sRNA arrays to reprogram metabolism. We've also done a lot of work on optimizing metabolic pathways from peripheral biodetoxification to increasing NADPH regeneration. And we've developed riboswitch biosensors for a various number of ligands of interest, ranging from neurotransmitters like dopamine, hormones like thyroxine, fluoride, which is something in our water, and DNT, which is an explosive, hopefully not in the ground. Of course, we have our web interface where people can go and use our software to design their genetic systems. We have a spin-off company that manages the maintenance of the software so that my students don't worry about it. It's just handled by by me and some other people. And finally, we've had a lot of people use our software to engineer a large number of different genetic systems with lots of applications, particularly in metabolic engineering. Okay, so today I want to introduce our new promoter calculator algorithm and explain how we leverage both biophysics and machine learning to come up with a free energy model for the interactions between RNA polymerase and segment factor 70 with promoter DNA sequences. Okay, and then I'm going to be introducing another new algorithm called the non-repetitive parts calculator, where we show how to design large toolboxes of non-repetitive genetic systems, parts and systems. Okay, already getting some chats. All right, I'm gonna save some of these questions for the end, just because they're overview questions. But okay. Wait for us. There it goes. Okay, you can see that. Okay. So just to start off with, here's a common question. Are promoters modular parts? So we have data to show that in fact, promoters are not modular genetic parts. The sequences upstream of the promoter, the sequences downstream of the promoter, particularly that's the messenger RNA, they can affect the transcription rate of the promoter by up to 10-fold. Okay, that's a pretty big number. And so when we think about modularity, we can maybe think about larger systems being modular, but I think we've come to a point in synthetic biology where individual parts that have rather short DNA sequences, we should not assume that they are modular because commonly they are not. Okay. And here, as an example, is the J23101 promoter. It is rather short, 40 base pairs long, and depending on the sequence upstream and downstream, again, transcription rates are changing by quite a lot. So how do we develop a promoter calculator, a biophysical model able to predict transcription rates for any DNA sequence, a sequence complete model? So to answer that question, we designed 14,206 promoter sequences that systematically perturb the molecular interactions controlling how well RNA polymerase segment factor 70 binds to promoter DNA. Okay, can you see my mouse right now? Okay, so so here we have the specific interactions controlling binding, and we've broken up those interactions according to their elements. And so we have the initial transcribed region, we have the discriminator, we have the -10 hexamer, the -10 extended, -35 hexamer, the up element, okay? For each term here, we quantify the interaction energy according to a binding free energy. And our goal is to develop a free energy model to predict the strength of each of these interactions. And in order to do that, we designed promoter sequences that vary that strength according to unknown coefficients. And our goal here is to figure out the coefficients controlling the strengths of these interactions. So we're changing DNA sequences, and we have an idea, hypothesis regarding how those DNA sequence changes will influence each interaction, but we can't a priori predict the actual interaction strength, okay? And so there is a strategy where even though you don't know what the outcome is, you know that you can change the outcome by a lot by suitably designing your DNA sequences. And then through the process of designing these sequences, constructing them, characterizing them, you could then figure out the interaction coefficients using machine learning. So that's what we did. And so here's an example where you have two different DNA sequences shown here. We are here just changing the -10 hexamer, and but you might notice that just changing, just making a modification to a promoter sequence can actually have a large effect. It can not only affect the transcription initiation rate of that promoter, it can also affect the transcriptional start site of that promoter. So in order to develop the promoter calculator algorithm, we needed to carry out both transcription rate measurements as well as transcriptional start site mapping measurements on all of our 14,206 promoter sequences. And so that's what we did. So we did TSS mapping and transcription rate measurements shown here. And you do notice that the TSS mapping measurements, it's a distribution. A single promoter sequence can actually give you more than one TSS, transcriptional start site. And depending upon which sequences we modified, we actually could accidentally shift the TSS location, okay? And then further, for a lot of our sequences, there are two predominant TSSs. And so there's an upstream one and then a further downstream one. And so this kind of gives you an idea that it can be difficult to design a promoter a priori with only one transcriptional start site. Just by random chance, you can accidentally introduce a promoter with two transcriptional start sites, even if you know what you're doing, okay? And further motivation for developing this model. But now that we have these transcriptional start site measurements, we can figure out exactly where the RNA polymerase bound, and that helps a lot during model development. And of course, we have the transcription rate measurements, and they make sense in terms of how mutating various regions of these motifs and their sequences affect how much change in transcription rate you're going to get. And so the -10 hexamer is very important, and the spacer region is very important, -35 hexamer very important, whereas the discriminator element and ITR do have a change, have an effect on transcription, but they're not as important. The other important bit that is very important to note is that we carried out all of these measurements in vitro reactions in test tubes. So there's really only 4 ingredients in this reaction. There's the RNA polymerase sigma-70 enzyme. There's all of our plasmids that we pooled together, and they're each molecularly barcoded with a different DNA sequence. There's NTPs and buffer, okay? And that means that when we are reading out the transcription rates from all these different promoter sequences, we can say very precisely and definitively that we're measuring the differences in binding free energy for RNA polymerase and sigma-70 to the DNA. There are no other transcription factors. There are no other initiation factors or elongation factors that are having an effect on our measurements, which is very important. And so then when we go to develop and formulate our free energy model, we have to keep in mind that we have already set up a competitive system. So we have our 14,000 plus promoter sequences that are all competing to bind to RNA polymerase and sigma 70. And there isn't enough RNA polymerase and sigma 70 to go around. So it's definitely a limiting reagent. And so that first step for recruitment is very important for controlling transcription rates. And we've set up a competitive process where the promoters that bind best to RNA polymerase and sigma 70 will have the highest transcription initiation rates. So that's the first step. The second step is that transcription is definitely a cyclical process. And so we're actually going to be recording multiple rounds of transcription taking place during our experiments. And these are not short assays. They're like 15 to 18 hour in vitro transcription assays. So there's plenty of time for multiple rounds of transcription to take place, which means that during model development, we can actually assume that we do not have any accumulation of intermediate states. And so during the, writing down the equations in this model, we come, to a point where we have to start making assumptions. We do assume that there is no accumulation of those intermediate states, but we've also set up the experiments to make that very true. And then finally, we pick one of our promoters as a reference promoter, and we carry out all of our comparisons to that reference promoter. So we have comparisons in the model calculations, and we also have comparisons in the measurements, which gives us one or really two simple equations to consider at the very end of the day. So you can go through all this model development and get to this point at the very end. And so we have measurements on the left, a log ratio between the transcription rates of our promoters as compared to the transcription rate of our reference promoter. So Tx over Tx ref. And that is going to be equal to negative beta times the difference in binding free energy between our promoter of interest and the reference promoter. And so here we really have a delta delta G. And so when we develop our model and make our predictions, we're really training our model on the delta delta G's, and we're making predictions on delta delta G. And that allows us to get around this idea of having proportional measurements. And then finally, we write down a free energy model for delta G total in terms of all those different interactions, and we add them up. Okay, now comes the machine learning, because we have a lot of data, okay? Not an infinite amount of data, but a lot of data. And we want to come up with a free energy model where we can directly convert our idea of what the biophysical interactions should be to the coefficients and strengths of those biophysical interactions. And so one way to do that is to use supervised machine learning, where we of course have labeled data and we have labeled features, okay? And those features might be numerical. For example, we can actually calculate the minor DNA groove width for the upstream up element. And we know that that minor DNA groove width will have an effect on the binding free energy, delta G up. But we don't exactly know what that effect is, but we know that the DNA's minor groove width has an effect, okay? And so we input a numerical feature, one for the distal site, one for the proximal site in the up region. And the goal of machine learning is to then tell us what the effect of the DNA minor groove width is on transcription rate. We can do the same thing with the spacer length, with DNA rigidity, and with the thermodynamic stability of the initial transcribed region. Those are all numerical features where we calculate a number and we plug the number into machine learning and we ask the question, what is the coefficient in front of this number that determines the binding free energy for that particular interaction? Then we have the categorical features. For example, we know that the -35 hexamer and -10 hexamer are really important, but we don't know the specific binding free energy for a particular sequence motif. Okay, so we don't know the coefficient attached to each one of these different motif sequences. And so what we can do is write down a list of all of the different motif sequences, and we have a choice as to how we broke up the motif. And so here we chose to write them down as for the -10, it's 2 3-mers. And for the -35 hexamer, it's also 2 3-mers, a 3-mer being a 3-nucleotide sequence, which means there's 128 possible -35 sequences, 128 possible -10 sequences. Therefore, we have 256 coefficients, okay, for describing the interactions between the -10, -35, and RNA polymerase / sigma factor 70. But the great thing about machine learning is that you can actually specify quite a few features, hundreds, thousands of features. And machine learning, if given enough data of good quality and good coverage, machine learning will be able to give you back the coefficients in front of each of these different interactions, which is what we did. So we carried out a rigorous procedure where we split our training, or all of our data, into a training set and a unseen test set. And with the training set, we then carried out 10-fold cross-validation in order to develop a very accurate but also highly generalized model. And to do that, we used ridge regression, which is a type of linear machine learning algorithm that uses regularization. Although we got the same result whether we use lasso or elastic net, which is other types of linear machine learning models with different forms of regularization. One second. And importantly, after we developed this model with the training set, we then applied the model on the unseen data set, and we got the same accuracy, which is exactly what you would expect to get if your model is generalized across your entire data set. Another way of seeing this is that you can plot the learning curve, which is the mean error of the model predictions as you increase the amount of training, the amount of data in your training set. And what you're really expecting to see is that as you increase the amount of data in the train set versus the test set, that the amount of model error should converge. You should get the same amount of model error in your model when evaluated on both the test set versus the train set. Okay, so that the model is giving you the same accuracy of predictions on both sets of data. And you want to see that convergence take place before you run out of data. And so the fact that we see this point of convergence between the train set line and the test set line means that we had enough data to develop our accurate and generalized model. We didn't run out of data. And then finally, and perhaps most importantly, what do you get after you've trained this model? With other types of machine learning, particularly neural networks, you just get a predictor, but you don't know how the predictions are taking place. But the way that we carried out our machine learning, coupled to biophysics and categorization, we can actually examine the coefficients that the machine learning has identified and come up with the free energy model that was developed and trained, and then go back and make sense of that free energy model to figure out if it really does match our expectations based on other experiments, other data sets, other labs, other observations. And so here are the coefficients for each of the features that we included in our machine learning model. And so we do have those 256 motif sequences, and I'm showing the top 20 and bottom 20 by interaction strength. And you can see that the consensus -35 is TTG ACA, and the consensus -10 is TAT AAT. And that's exactly what the machine learning pulled out as the motif sequences with the highest interaction energies, the strongest, which is here the most negative. And then importantly though, the machine learning was also able to fill in the gaps, not just the consensus motifs, but all possible motif sequences, and assign numbers to all of them. And we did the same with the discriminator, with the up element, the spacer sequence and its length, and the ITR thermodynamic stability. So for each of the features that we included in the machine learning model, we're able to figure out how they contributed to the binding free energy, and we get predictions out of it. So we learn as we predict. Okay, so we figured out that those predictions work very well, even if we do not provide the transcriptional start sites into the model. So just to keep in mind, when we initially trained the model, we did have the transcriptional start site mapping data in order to develop a really good model. But then later on, if we take away the transcriptional start site data and we carry out a classic stat therm approach where you scan across the DNA, you enumerate binding states, you calculate energies, and you find the energies that are the most that are minimum, and that will tell you where RNA polymerase is going to bind. Once you go through that process, you can actually make predictions without having the TSS information. So that's step one. And how well are those predictions? They're basically the same accuracy as during model training. So that's basically a validation of statistical thermodynamics. Next, we then compiled data sets from other labs as well as our own, and we ran the promoter calculator predictions on those data sets, and they have different characteristics. So in one data set, we have thousands of genome integrated promoters that are inside of cells, and therefore they're at 1 to 2 copy, and they've but of course there's other transcription factors, other sigma factors that are at play affecting the transcription rates from these promoters, and yet the promoter calculator can still accurately predict their transcription rates pretty well. And then we have another data set where we have, again, another 4,000 plus promoters that are now on plasmids, okay, multi-copy plasmids inside of cells. And we do see a lot more noise in the predictions versus measurements, but we know ahead of time that because these promoters are on multi-copy plasmids, there's going to be more variability in DNA copy number, and therefore more variability in mRNA level, and that could be a source of the confoundingness of these predictions. Okay, so again, going from left to right, we have on the left we have in vitro measurements, very clean, very precise. On the right, we have messy multi-copy plasmid measurements or messier, and we do see a reduction in the model accuracy. And then, importantly, can we use this model to make predictions during rational design? And the answer is yes. So we can of course use computational design with the model, and we can design promoter sequences with targeted transcription rates. We designed 16 different synthetic promoters, making sure that all of their sequences were very dissimilar. And we can see that, yeah, we were able to accurately predict the transcription rates of those 16 different promoters across a very wide scale. Okay, so that is our new promoter calculator. How am I doing the time? I got 20 minutes left. Awesome. Wait, right, it's sort of at 2, yes. All right, so I'm gonna, normally I'm gonna probably do the same thing here. I'm gonna just very quickly show that over the years, we've made successively better versions of the RBS calculator, taking into account more non-canonical interactions. Right now, we're at version 2.1. And that's very common. You start off with the interactions that have the most effect, and then you progressively incorporate interactions that have lesser effects, but over time, you can get better accuracy in that regard. I want to highlight an interesting biophysics question here. Which happens first? Can the ribosome bind to a messenger RNA before the messenger RNA folds into its structure? This is not a thermo question. This is a kinetics question. Okay. And so this question really arose when we started to study the errors in our model predictions. And we noticed that messenger RNA sequences with very long structures had a lot more model error from the RBS calculator predictions. Okay. And that really implied that, of course, there was something special about those long messenger RNA structures, perhaps because they took a lot more time to fold. And so we proposed a model, a non-equilibrium model of translation initiation called ribosome drafting, whereby you can actually have a scenario where the ribosome binds to a messenger RNA before it folds into a structure. And if that takes place, then another ribosome, a second ribosome, can then bind to that same messenger RNA right after the first ribosome, and thereby prevent that messenger RNA from ever folding into its structure. And we call it drafting, just like a Peloton, bicycle drafting, or NASCAR drafting. It's a same idea that if you have a multi-unit effect, you can save some work. And so the ribosomes here are absolutely saving themselves some work by binding successively to the messenger RNA faster than the messenger RNA's refolding rate. Does this actually take place inside of cells? How do we figure that out? So we computationally designed fast folding and short folding RNA hairpins, but importantly, we designed them so that their folding free energies were near identical. So if only thermodynamics was at play, then the translation rates with these messenger RNA structures would be identical. But if kinetics were important, then the translation rates would be greatly affected for the fast folding versus the slow folding hairpins. And so we introduced these messenger RNAs, hairpins, into constructs, and then we measured, and we figured out that yes, indeed, a messenger RNA with a slow folding hairpin can actually have ribosome drafting take place. It will have an acceleration, an increase in translation well above the equilibrium prediction. And we actually can carry, we did carry out a whole successive set of experiments. We varied the ribosome's binding rates. We varied the kinetics of RNA folding. And we, in comparison to a non-equilibrium Markov model, we expect a sigmoidal relationship with the slow folding hairpin. And that's exactly what we see. So we see that with the slow folding hairpin, we only see ribosome drafting if the ribosomes can bind fast enough to bind faster than the slow folding hairpin. If the ribosomes bind slow, then it doesn't matter if the hairpin was fast folding or slow folding. Okay, that was an abbreviation of that story. Okay, for the next 5 minutes, I'm going to talk about our new model of messenger RNA decay. Okay, we often don't think about messenger RNA decay, but it does have a huge effect on gene expression levels. So it is a complicated process to study because there are so many interacting proteins and enzymes that work together to degrade a messenger RNA. And so you have transcriptional elongation and co-transcriptional folding of that RNA. You have translation initiation, ribosomes binding to the messenger RNA as it's being transcribed. And those ribosomes are actually protecting the messenger RNA from degradation. You then have enzymes like RNase E that bind to the 5' end of the messenger RNA and start chopping. Okay, but they chop their endonucleases. So even though they bind at the 5' end, they still chop in the middle of the messenger RNA. And there is an enzyme called RPPH, which will dephosphorylate the 5' end of the triphosphorylated messenger RNA transcript. And through that dephosphorylation, it can increase the ability of RNase E to bind and cut. RNase E doesn't have to bind to the 5' end. It can also bind pretty much anywhere along the messenger RNA transcript, and that is called direct entry. So the question here is, how fast does that happen? And then there are, of course, many other RNases, like RNase 3, that bind to specific structures. So if you have a structure that forms in your transcript, RNase 3 can bind and cut it. And then the question is, what happens after that? So in a learn by design approach, we figured out the rules controlling messenger RNA decay. And so we have a pretty simple equation for that tells us how single-stranded RNA regions in the 5' UTR affect messenger RNA decay rates. And so the longer the single-stranded RNA region, the easier it is for RNase E to bind and chop. And once RNase E binds and chops, that's it. That messenger RNA will very processively be chewed back, and you won't get any more protein from it. And so on the left side are messenger RNA level measurements. On the right side, we just simply invert those measurements, and that tells us decay rates. And so you can see as we increase the amount of unstructured RNA, starting at the small scale, so lengths from 2 to 20 nucleotides, we get approximately a linear relationship between unstructured RNA length and decay rate. And that's what we have for a simple equation up here, a linear relationship between length and decay rate with a footprint of 2 nucleotides. So you need at least 2 nucleotides for the RNase E to bind and cut. But then we found that after 20 nucleotide length, that we see an attenuation effect, okay? And we wondered, well, why? Why do you get a lower degradation rate as the length of single-stranded RNA goes above 20? And then we started to think three-dimensionally and think about loops forming in the messenger RNA. Because if you have a single strand poly A sequence of messenger RNA, eventually it's going to form a loop. And we know the persistence length of messenger RNA, particularly for poly A sequences, is around 20 nucleotides long. Not exactly, but close. And so all we need to do to predict this effect is drop in the probability that a messenger RNA sequence that is unstructured does not form a loop. And that is exponential negative L over P, where P is the persistence length of that RNA. So the larger the P, the less likely that it will form a loop, okay? P does depend on sequence, and so it does vary depending on the messenger RNA transcript, but at least for poly A's, we're pretty close. So the red dashed line is including that loop formation effect. Next, we do know that the ribosome can protect the messenger RNA from degradation, but how? How much? We don't know. And so therefore, we carried out a series of experiments where we systematically varied the translation rates of our messenger RNAs, keeping the 5' UTR otherwise the same in terms of the amount of single-stranded RNA. And we measured the amount of mRNA level with RT-qPCR. And of course, we see that as we increase the RBS's translation initiation rate, we get an increase in fluorescence levels. We also get an increase in messenger RNA levels. So we have to figure out that effect. If we take the fluorescence levels, divide it by the messenger RNA levels that we both measured, we do see a linear relationship between predicted translation initiation rate and that ratio that is measured. So the RBS calculator is working appropriately in this regard. But then importantly, if we compare the decay rates of that messenger RNA versus the predicted translation initiation rate of the for the RBS, we see an inverse relationship. So higher translation initiation rates gives us lower decay rates. Okay, so we're quantifying the ribosome protection effect. How do we make sense of that? How do we actually predict the ribosome protection effect? And so here we utilize results from a model called TACEP, which stands for totally TACEP. Oh boy, I forgot off the top of my head. But it's a multi-particle with footprint model of the movements of ribosomes along the messenger RNA transcript. And so you have the distance between adjacent ribosomes. That is a very key important variable that we want to know from this model. We also can input in the ribosome's footprint, as well as the ratio of the ribosome's initiation rate over its elongation rate. So based on just these three numbers, we can, or really based on these two numbers, alpha and F, we can calculate the density of ribosomes on the messenger RNA, and we can calculate the distribution of distances between adjacent ribosomes. And we can calculate the average distance between adjacent ribosomes. And it's that average distance between adjacent ribosomes that we hypothesize would directly control the mRNA's decay rate. If two ribosomes are really far apart, they're leaving a lot of room for RNase E to come in and chop, chop, chop. Whereas if the D is very small and the ribosomes are super close together, there's no room for RNase E to get in there and start chopping. So high D, high decay rate. Low D, low decay rate. And that's exactly what we see. So we go through this and look at the average distance. We then compare the average distance to our messenger RNA, to our mRNA decay rate measurements, and we see the sigmoidal relationships. Okay, and they're pretty good, again, for the number of data points that were collected. Now we have more data on the pipeline. We recently completed a study where we constructed and characterized 62,000 different messenger RNA transcripts, and we measured the mRNA decay rates using rifampicin treatment, rifampicin, to stop transcription. And so from that, we're able to then fit exponential curves to decreasing, messenger RNA levels and get their half-lives. And so those half-lives range from, very short, so sub 1 minute, to very long, let's say just about 20 minutes. Okay. And we're still, chewing on this data and developing predictive models with this data set. This is one example. You can throw all the data into LightGBM, which is a decision tree, random forest, or really it's a gradient boosting model, but uses a type of random forest. And you can plug in all of the biophysical characteristics that we think are important to controlling RNA decay. And based on these features and the data set, you are able to develop a pretty good model to make predictions. But again, we're still chewing on this. Okay, so I'm gonna stop there. I think that's good enough. The second part of the talk is published, and I just want to highlight this paper. So you can go over to Nature Biotech and read all about the automated design of non-repetitive genetic parts and how we constructed and characterized thousands of non-repetitive promoter sequences that vary transcription rates across 820,000 fold that you can use all at the same time, for example, in a synthetic genome, without introducing repetitive DNA, therefore improving the evolutionary robustness of your genetic system. And then one application of that is engineering extra long sRNA arrays that is expressing many CRISPR sRNAs at the same time. And this is also published in Nature Biotechnology. And here we demonstrate that you can actually express 20 CRISPR RNAs at the same time without triggering homologous recombination. And by doing that, you can then redirect metabolic flows, and you can overproduce a desired chemical of interest, just as a demonstration. So with that, just to finish up the talk, we need to develop an engineering discipline for biology. I'm simplifying, but it's 3 steps. We need sequence complete biophysical and system level models, design rules and optimization algorithms to engineer systems with desired criteria and constraints. And we need to think about the, holistically about our whole system, which is not just the organism, but everything surrounding the organism as well. And we need to engineer everything to achieve our desired objective, whatever that objective happens to be. And in order to do that, we need to develop a mature, discipline. So I'd like to thank my graduate students, particularly Ayan Hussein, Travis LaFleur. Travis is responsible for the promoter calculator. Aaron, Harry, Morgan, and James, as well as alumni. Alex, developed the extra long SGNA array calculator alongside Sean Halper. Grace also developed, we have some other examples of developing new RBS switches. Okay, and with that, I'd be happy to answer any questions.
Excellent talk, Dr. Salis. I had a question about ribosome drafting. Do you think it's facilitated by the interaction of RNA polymerase and the ribosome through one of those ribosomal proteins that's supposed to do that?
So in E. coli, there is a factor, NUSG, which is responsible for coupling together the ribosome with RNA polymerase. And there's thought that that coupling exists to keep, actually prevent RNA polymerase from getting away from the ribosome, so that you don't have too much unprotected RNA that could then be degraded. Now, that interaction is not universal. It does not take place in Bacillus subtilis. But again, Bacillus subtilis does have pretty distinct process for its messenger RNA decay. Some differences in its enzymes. So I would say that that we could call that the pioneering round of gene expression. But after that, that's when ribosome drafting is at play. Because again, it's a non-equilibrium phenomena. The ribosome can actively unfold RNA structures. And as soon as a ribosome binds, unfolds that structure, and then a second ribosome can get in there before that RNA refolds, well, that's a non-equilibrium process. It doesn't matter if that takes place at the pioneering round or the second or third round. It can happen at any time.
Interesting. We have a question in the chat.
Can you predict the success probability of companies or their symbiotechnology using our potential program? Okay.
That's my question. And
Yes.
I know the Howard is one of the genius I know. So I can ask any question, any challenging question. So
Okay.
I'm asking.
All right, very good. How about this? If you're throwing darts at the dartboard, do you want to have a steady hand? Or how about this? Would you rather throw darts that are at the dartboard that is 3 feet away or 100 feet away, right? How many darts do you need to hit the bullseye, right? So the goal is that if you want to engineer a sophisticated genetic system, you cannot do it without rational design. You can't, you can try to say, oh, I'm going to build a gigantic library. I'm going to develop a high-throughput biosensor. All those different shortcuts to achieving the goal, they fail once the system gets too complicated with too many interacting components, or the genetic system just gets too big and unwieldy.
Sure.
And so we as scientists and engineers, we love to get really creative with all these shortcuts. And we're often really good at doing that. But then thinking long-term, those shortcuts don't have staying power.
Sure. So I asked the question because I mean I met one technology company actually doing sort of the technology success rate kind of prediction algorithm developing. So but my real question is I just wanted to hear. This is kind of because I mean all the technical thing I already read your paper, so I don't have any additional question because that paper is already explained everything. So my question is actually Ginkgo's future, because my worry is they giving lots of promise to us, but at the same time they are my friend at MIT. But at the same time I also heard many concern about the company, because if they fail, they will have the collateral damage to synthetic biology community. So I just wanted to hear from you, because you are one of the pioneer, and I wanted to hear your opinion on the company.
Well, are there any short sellers in the crowd? I mean, I think I mean, how about this? It's still too early to judge the whole field in terms of our commercial success. I will say that. Is it too early to judge one company over another? I don't know. I mean, you always hope for more success.
Sure.
But you also don't know what are the other constraints that people are dealing with.
Right, but the Ginkgo is kind of unique because they do not produce anything, but they has the technology, unique technology or the concept.
Yeah. How about this? Ginkgo does use our software, but not everybody at Ginkgo uses our software. Right. So I don't have any interior knowledge of what they do, like their success rates internally, but I would be personally interested in seeing the numbers on people. How about this? Not specific to Ginkgo. Have people who use our software and universities and labs that use our software, they tend to publish more on stuff that works really well.
Sure.
Versus others that they still publish, but it's always like a screening thing, and at the end of it, they get one or two things that work well. Does that make sense?
Yeah,
or
I use
a lot of money, and
And yeah, I need to go. But good to chat with you. I love to see you, but hopefully in person near future.
Okay.
Thank you so much. Your brilliant talk as always.
Thank you very much. Okay, would these systems be able to predict, say, expression of a specific protein in multicellular eukaryotic organisms? Okay, this comes up a bit, Pratima. So these biophysical models are specific to prokaryotic organisms, which does include mitochondria and chloroplast, because of course, those organelles use prokaryotic gene expression. That doesn't mean that the biophysics that we have, that I've been discussing, of course, those biophysical interactions are similarly taking place in other organisms. You would just have to use that knowledge to reformulate models for those other organisms. We have started to go in that direction. We have an early model of transcriptional initiation for yeast promoters, but it's not a predictive model. It's more like an explanatory model. But that's also in that 2020 Nature Biotech paper. And then Rami, you have a question as well.
Thank you. I don't know, do you hear me?
I can't hear you.
Yeah, thank you. So I was wondering on the in vitro transcription assays, whether you detect bidirectional promoters or transcripts running.
I can barely hear you. Can you get closer to the microphone?
Yeah.
Hold a sec.
Worst case scenario, just type it in the chat box.
Do you hear me now? Is it any better?
It's very, gotta crank that up.
It's on max, but I don't know. Okay, I write it in the question.
Oh, okay. Actually, if I turn up my volume to the highest level, I can hear you.
Apologies, it's on max on a red level, but I was wondering if you observe bidirectional activities from the promoters you use in the in vitro transcription.
Yes, so we do know that antisense RNA transcription takes place in our systems. We can use the promoter calculator to make predictions both on the forward strand and the reverse strand in terms of transcriptional direction. And we actually use those predictions in our designs to make sure we don't accidentally introduce cryptic promoters going in the antisense direction, which we have uncovered in some recent projects, which mess up lots of things. But then there are other times when you want to, let's say, reduce the leakiness of a promoter of interest. And one strategy for doing that is to purposely introduce an antisense promoter to produce antisense RNA that binds to the messenger RNA and degrades it. And in that way, you can actually reduce the overall leakiness in transcription rate from the forward promoter. So, forward promoter, reverse promoter. If this is inducible or going from high to low, but it's still leaky in the off state, you can actually increase the antisense promoter so that it degrades that lower level of transcription in the off state.
From a given promoter, if you were to remove so let's say you have a forward activity and reverse activity, if you could, from the sequence level, eliminate the reverse functioning promoter, does that influence the forward activity as well?
It can, because particularly if you have, let's say, a promoter region that is bidirectional, and RNA polymerase, it's going to bind on opposite strands. And so if you remove the reverse, if you modify the sequence, keeping the transcription rate in the forward direction the same, removing the transcript or lowering the transcription rate in the reverse direction, you basically are allowing RNA polymerase to only bind in the forward orientation, and it could actually more likely do that. And so that goes back into competitive binding. We're not sure yet, but one hypothesis is that RNA polymerase can't bind to both of the strands of a promoter region at the same time, which is probably true, but it's something that we need to test. Just looking at a crystal structure, it's unlikely to take place. We still have lots of remaining questions. For example, if you have a larger promoter region and you have multiple places where the model is predicting that you have high transcription rates, what are the actual transcriptional start sites and transcription rates? It could be an additive, or it could be there could be a level of competitive binding. If the footprint of RNA polymerase is large enough to prevent multiple RNA polymerases from binding to nearby sites.
Yeah, I guess building off of that, what are your thoughts on how the field might approach prediction of transcription initiation rates, accounting for like the DNA binding proteome and nucleoid-associated proteins?
Yeah, I mean, we do have a funded project in that area, so we're going to be looking at that.
Awesome.
But molecular biologists have been long fascinated by chromatin-associated proteins like HU, IHF, so on, Fizz. They're very plentiful, or some of these are very plentiful in bacteria, and so they are glomped on pretty somewhat non-specifically, but they serve an important role. And the key question is, what is that role? Do we have to design our promoters differently to either attract them or repel them? We do know in the eukaryotic world, we have some experience designing promoters so that you have a nucleosome-free region, so that transcription factors combine in an accessible fashion, and that there is a high affinity +1 nucleosome binding site right near the transcriptional start site, which is a key requirement for eukaryotic transcription. So what are those corresponding rules in the prokaryotic world? I think that's very understudied.
Any other questions from the audience? I have one more. Do you have any thoughts on like what the approach might look like to make, say, promoter calculators for other species, other bacterial species?
So keep in mind, all of those initial measurements that we carried out are in vitro. And so we expressed RNA polymerase and sigma factor 70 they're in complex. We add them to a test tube. We ran our in vitro transcription assays. We then carry out our measurements on the products from that reaction. You can do that with any organism, prokaryotic. You can do that with any sigma factor. Yep. So again, another part of a funded program project.
I look forward to all the work that comes out of your lab.
Thank you. Okay, very good. I think we're
All right, yeah. I think that'll end this session. There's no talks afterward.